Lab Splaining. We're us scientists here at InnerPlant, explain some of the amazing science we do, for those of you who don't work in a lab. On this episode, we're going to learn about Polymerase Chain Reaction, or PCR for short.
In simple terms, PCR combines the search function, like in a computer, with the copy and paste function. This allows us to make millions to trillions of copies of small pieces of DNA for testing later.
So how does PCR work? Well, it starts by heating the DNA, which separates the two strands in a process called denaturation. Once the DNA strands are separated, a small molecule called a primer is added, which binds the specific section of DNA we're looking for. This process is called annealing.
The third and final step is known as extension. This is where we'll add a heat-stable enzyme known as polymerase, which binds to the spot that the primer has located. The polymerase then builds along the DNA template to create new DNA strands. This is the copy and paste function, which is repeated many, many times.
With each cycle, the amount of DNA doubles. So even after just a few cycles, we already have millions to trillions of copies of the specific DNA we're looking for. This is really important because it allows us to do downstream testing and identify genes of interest.
At InnerPlant, we use PCR for grabbing promoters from plants that we use, creating new sequences of DNA, and checking to see if a plant has our DNA trait.
I hope you've enjoyed learning about polymerase chain reaction, or PCR for short. If you have any questions, or if you have a topic you'd like explained, just drop it in the comments below. Happy sciencing!